Meiosis is a type of cell division that results in four daughter cells, each with half the number of chromosomes of the parent cell. This process occurs in two different phases, meiosis I and meiosis II. First, let's start with some vocabulary. This is a chromatid. A chromatid is one strand of genetic information, otherwise known as DNA. The yellow bead in the middle is a centromere. A centromere is what scientists use to count the number of chromosomes. So this one chromatid is equal to one chromosome. Next, we have sister chromatids. Sister chromatids are identical copies formed by DNA replication with a common center, the centromere. In this slide, we see two sister chromatids, and again, this is only equal to one chromosome because of the centromere in the middle, which is represented by the yellow bead. This is a homologous pair, also known as a tetrad. A homologous pair has one chromosome from the mom and one chromosome from the dad in humans. A chromosome is a thread-like structure that contains genetic information. Before meiosis I can occur, a process called interphase occurs. In this process, the one chromatid folds over and its DNA is replicated to form two sister chromatids, also known as one chromosome. Then, one chromosome from the mom and one chromosome from the dad pair up to form homologous pairs. Now that homologous pairs are formed, prophase I of meiosis I can occur. Prophase I begins with a diploid cell, meaning there are three homologous pairs. During late prophase I, the homologous chromosomes come together to form tetrads. In prophase I, crossing over also occurs. Crossing over is when bits and pieces of the homologous chromosomes are exchanged, which produces a new combination of alleles on each chromosome. First, this process starts with the chromosome tetrad. Then, we see the arrow pointing to the pairs crossing over, and finally pieces are exchanged to form the new combination of alleles. The next step in meiosis I is metaphase. Metaphase is where the paired homologous chromosomes line up across the center with their respective pairs as shown in this picture. And then, as the cells move into anaphase I, the homologous pairs of chromosomes separate. During anaphase I, spindle fibers pull each homologous chromosome pair toward the opposite ends of the cell, and the separated chromosomes cluster at opposite ends of the cell. After anaphase I, cells undergo telophase I, in which nuclear membranes form around each cluster of chromosomes from anaphase I. The red outline shows the nuclear membranes forming around the two clusters. Then, cytokinesis follows, in which two new cells are formed. The nuclear membranes separate and each form their own cytoplasm. The new cells formed have sets of chromosomes and alleles that differ from each other and from the diploid cell that enters meiosis I. Now, finally, meiosis I is complete, but meiosis II still has to occur. The first step in meiosis II is prophase II. In prophase II, the sister chromatids become more distinct. This means that you can easily tell which chromosomes are in the two new cells formed from telophase I and cytokinesis I. Metaphase II and anaphase II are exactly the same as metaphase I and anaphase I. The paired homologous chromosomes line up across the center of the cell, and then the spindle fibers pull each homologous chromosomes to the opposite ends of the cell. Here we can see the spindle fibers pulling each homologous chromosomes to the opposite ends of the cell. This part of the process is called anaphase II because this occurs during meiosis II. So far, meiosis I and meiosis II are very similar to each other. However, this changes in telophase II. During telophase II, four haploid nuclei are formed instead of a nuclear membrane forming around the cluster of chromosomes. Then, after telophase II, is cytokinesis II. The division of the cytoplasm in cytokinesis II results in four haploid daughter cells. These haploid daughter cells are known as gametes. Gametes are the male or female sex cells, depending on the gender you are. And voila! This is the whole entire process of meiosis I and meiosis II in order to get four haploid daughter cells. But wait, there's more. There's an essential question that needs to be answered before we finish up. What is the relationship between meiosis and Mendel's model of inheritance? The answer? Mendel created three principles: principle of dominance, principle of segregation, and principle of independent assortment. All three principles are represented within meiosis. Dominance versus recessive genes are exhibited when there is a crossing over of chromosomes in prophase 1. Segregation occurs as spindle fibers pull each homologous chromosome to the opposite ends of the cell. Finally, the independent assortment of genes occurs as homologous pairs separate through meiosis 1, resulting in haploid cells that have a random independent assortment of chromosomes. Now, we have successfully accomplished, or at least I hope we have, in explaining meiosis and its connections to Mendel's model of inheritance. I hope you have enjoyed this video and have become more educated in the world of meiosis.